Darwin had trouble with the fossil record. The expected progression of forms could be seen in a general way over the broad sweep of geologic time, but the fine intergradations that his theory seemed to demand weren't really evident. He explained this mainly by the incompleteness of the fossil record. Of course, with 150 years of further collecting and analysis, we now know the fossil record, though certainly imperfect, is actually pretty wonderful, at least for some groups. As Neil will discuss this afternoon, many gaps between the major groups have been filled by beautiful intermediates. And as Doug discussed yesterday, individual species may follow random walks or show stasis and punctuations, but rarely show the continuous transformation over millions of years. And there's nothing anti-evolutionary about that. It's just a different dynamic from the simplest possible model. How that dynamic adds up to long-term trends is a different talk, which I'm not going to do today. But these uses of paleontological data brings me to the theme of my talk, the renewed partnership between evolutionary biology, or more precisely, evolutionary neontology, and evolutionary paleontology. I'm not going to go over past debates, of which there have been plenty. Instead, I want to talk about some areas where the fossil record can serve as a laboratory for testing evolutionary hypotheses that are difficult to resolve any other way, and in some cases, where the lack of a direct window into deep time can bring actively misleading results. One of the great challenges is to generate methods that can leverage even poor or spotty fossil records to rule out hypotheses, and I'll say a few words about that along the way. But the first step is to develop method and theory using a group that is both well represented in the modern world and has a robust fossil record. For that, I'll focus on marine bivalves, scallops and mussels and cockles and oysters and so on, which are becoming a model system for the integration of paleontological and neontological data and methods to gain fresh insights into evolutionary processes. So I'll be somewhere in between categories 4 and 5 of Jerry's scheme all the way through this talk. The fossil record provides unique data on space, time, and form. Regarding paleontology's time perspective, it provides access to raw rates of origination and extinction, to unique events like asteroid impacts or the origin of feathers, and most importantly, to long intervals that allow us to determine empirically the limits of extrapolation from short-term observations from short-term observations to long-term outcomes. Raw rates of origination and extinction, as opposed to net diversification rates, give direct access, for example, to the mechanistic hypotheses that underlie the net rates that are available for neontological data. For example, where species richness is tied to a time-calibrated phylogeny, as Bob Ricklef, Stan Roboski, and others have shown, the best model fits to molecular lineage through time plots are almost always involve unrealistically low, sometimes zero extinction rates, which is not realistic from paleontological data. Paleontology's spatial perspective is also important. Yesterday, Jerry was suitably derisive about assumptions of spatial stasis among populations and species. And that's true from a more macroevolutionary perspective, too. But fossils, even rare fossils, can constrain hypotheses and suggest new ones. And finally, regarding form, it almost goes without saying that the fossil record is really the only way to observe directly the timing and nature of morphological transitions and diversifications. Most of the most basic molecular toolkit for making giraffes, giants, squid, and monarch butterflies was apparently present in their worm-like common ancestor. Phenotypic inferences based on the phylogenetic distribution of regulatory gene networks can only go so far. Neil will talk about transition, so I won't step on his story, but the last part of my talk will involve a new way to look at diversifications that shows how a family of models are required to account for the dynamics that we see in the fossil record, and probably in modern groups as well. So, extinction is a real problem for strictly neontological data, of course, and if it's one thing the fossil record has, it's the trail of mayhem, destruction, and heartbreak leading up to the present fauna. There's significant variation in extinction intensity over time and among clades. But most neontological work is forced to adopt simplifying assumptions, as I mentioned, of constant extinction rates through time and among clades. And neither assumption holds, whether on the million-year time scale or on the 100 million-year time scale. And there's a huge range of data I could draw on to show this, but I'll use two examples from some work in progress. First, I probably don't need to belabor this point, but there's been huge variation in extinction intensity over time, and here's a time series for bivalve genera over the past 200 million years, from the Jurassic on the left to the present day on the right, with the big end Cretaceous or KT extinction as the big spike 65 million years ago. So that settles that. Now, the phylogenetic signal has been harder to quantify. But we've recently made some headway, and this is a molecular tree based on 28S, 18S, and CO1 sequences for 45 extant families of bivalves. There's a rough time scale at the bottom, but it's not important here, nor are the names along the side. What is important is the size of the circles next to the names, which are proportional to extinction rates calculated from the fossil record. And we omitted some families that failed to meet a minimum number of extinctions and taxa known to have a poor or uneven fossil record. But not only is there clearly differential extinction among clades, in fact, they range over an order of magnitude, but the extinction clusters phylogenetically, significantly, as shown by the red circles and larger groupings too. So here's a time series of phylogenetic clustering metric for our data on bivalve genera within families over the last 200 million years. Now the curve is not extinction, but how clumped it is in phylogenetic terms, from the start of the Jurassic on the left to the present day. The shaded bars are the 95% confidence intervals for the null expectation of no phylogenetic signal, with the dashed line showing the 0 value, the dashed horizontal line. The different widths represent the different durations of the intervals, the different heights, confidence intervals around that 0 mark for each bin, and the gaps represent times with insufficient data for robust confidence limits. There just weren't enough extinctions in that particular bin. Each named point, the ones that poke above the confidence bars, is a time interval that's significantly outside those confidence intervals. The big spike labeled Maastrichtian is again the end Cretaceous mass extinction, which removed more than half the genera globally. Looking over that entire time series, 81% of the 26 intervals where we had sufficient data show phylogenetic clustering, way more than expected. When we compare individual time intervals to the null expectation for its interval, based on the randomizations, 8 out of the 26 show significantly positive clustering. And no interval is significantly less clustered than random. And note that the Plio-Pleistocene boundary, way at the right-hand time, just 2 million years ago, falls way outside the confidence interval. So assumptions of constant extinction over time and across phylogeny are strongly violated. The fossils say it isn't so. And as Dan Roboski noted recently, this undermines many analyses, reinforcing Phil Ward's point yesterday about the need to be very careful about modern assumptions and making these large-scale evolutionary analyses. Now, another problem with diversity histories estimated from molecular data alone is that they're constrained to show monotonic increases, so that interpreting estimates of speed-ups and slowdowns can be problematic because most diversity-dependent mechanisms should involve the total clade, and perhaps other taxa as well, and not just the extant subset. But consider the diversity history of cockles, the cardiid bivalves, shown here from the beginning in the Mesozoic, 250 million years ago on the left, to the present day on the right, with the end Cretaceous, or KT extinction, 65 million years ago at the vertical line. The drawing, by the way, is by Ernst Haeckel. So the black curve, the higher curve, is the total diversity of the clade in a series of 5 million year time bins. And the red curve is the diversity of extant genera, those that make it all the way to the present day for the same time bins. Remember, that red curve is not a lineage through time plot. Those data will be a long time coming, though we're working on it. But we see some interesting contrasts. Superficially, the curves may look fairly similar, especially in the diversification following the KT extinction. But if we're trying to capture the dynamics, the devil is in the details. The basic trajectory of the extant taxa is a moderate diversification after the extinction, kicking into a higher gear about 30 million years ago. There's a slight hint of sinuosity to that high rate curve, but it's not significant at this scale. In contrast, the full clade shows a rapid diversification right after the extinction. It plateaus, perhaps in the late Cretaceous or in the late Cenozoic, or at least levels out, maybe as climates are becoming harsher and the tropics are narrowing. And there's even a suggestion of a drawdown over the past few million years. Because the modern seas are better sampled than Miocene, sampling biases go in the opposite direction from that observation, so it's probably real. So we get a qualitatively different picture from the two data sets. And assumptions, again, of constant and evenly distributed extinction rates are probably not an adequate response to these problems. And scallops show the same kind of pattern. This is just the Cenozoic, 65 million years ago to your left, present day on your right. And there are 2 points to take away here. First, the total diversity, the darker, higher curve, shows a roughly logistic dynamic leveling off in the late Cenozoic and then dropping down over the last 5 or 10 million years. the living taxa alone show a more continuous increase over time. And there are two interesting points here. Besides that difference in the qualitative trend, the second point is that you'd expect if they were, that the living in total diversity tracks would converge as they got closer to the present day. After all, the living taxa are just a subset of the total, but as highlighted by the red arrows, they actually diverge in the middle of the Cenozoic. There's a lot of diversity that expands and then goes again before we get to the present day. For some questions, that's perfectly fine, but if we want to talk about the ecological and biogeographic context of those living lineages over the past 5 or 10 or 20 million years, we're missing an important part of the story. But just so you don't get the impression that all bivalve diversification is logistic, with striking divergence between living and total diversities, here's a plot of telinid bivalves, a major group of bivalves, showing impressive concordance between extant and total diversity. So we really need to know the dynamics on a clade-by-clade basis. And of course, what we really need are new approaches that use incomplete data to constrain models. For example, perhaps models could incorporate the proportion of living and extinct lineages in a few exceptional time bins, living in extinct lineages of birds in the Eocene mesial deposits, for example, or ants in Baltic amber. And even those small bits of data can significantly constrain general models. Now, regarding space, my next topic, the fossil record is full of surprises, and one of my favorites is this fossil from the Oligocene about 40 million years ago in Germany. It's a stem group hummingbird, a group now known only from the New World. Extinction and rain shifts have distorted the biogeographic picture of this clade. And the fossil record is very rich in occurrences outside the present ranges of clades. Even omitting those that simply reflect climate shifts like palm trees in Greenland, I quickly ran out of room on this slide with ratites in the northern hemisphere, peccaries in the old world, rhinos in the new world, and many, many more. Now, there are methods that can, in favorable circumstances, make reasonable inferences on range shifts and centers of origin using biogeographic phylogenetic data on living species, but conditions aren't always favorable. And it's clear that we can't assume that lineages evolved or diversified in the regions we find them in today. And it's increasingly clear that many global events actually have spatial structure and dynamics, and these include the radiation of marine metazoans in the Ordovician period, which was the follow-on from the Cambrian explosion, the marine Mesozoic revolution with the rise of shell-crushing predators like crabs and specialized fishes, and so on. And there's the origin and maintenance of the latitudinal diversity gradient, the huge taxonomic and morphological diversity rise from the poles to the tropics. And this pattern has been known since before Darwin's time, but the dynamics in terms of origination, extinction, and invasion have been difficult to resolve, partly because of this problem that lineages don't necessarily reside in the places where they originated. So the debate has raged for a long time whether, as Stebbins put it, the tropics are a cradle or a museum of diversity. But the fossil record of marine bivalves, at least, show a fairly clear pattern. The tropics are both cradle and museum. This is the out of the tropics model we developed in 2006. Lineages preferentially originate in the tropics on the left as young genera. So the tropics are a cradle. As they expand outward to the right into higher latitudes without losing their tropical members, so the tropics are a museum, they build up diversity in the tropics while trickling up into the higher latitudes. The tropics are a museum not because they attract invaders from higher latitudes. In fact, that seems to be largely a one-way street, but because they accumulate homegrown diversity, older genera in this case, over time. And the fossil record of marine bivalves let us track this dynamic directly. So here's the proportion of evolutionary lineages, usually ranked as genera and subgenera, that first appear in tropical versus higher latitudes for 3 recent slices of geologic time. The youngest, the Pleistocene on the left, then the Pliocene in the middle, and then the late Miocene on the right. And as you can see, for each time slice, there's about a 70-30 split between tropical and temperate and extratropical originations. And once clades originate in the tropics, they tend to expand into higher latitudes. And this dynamic also occurs at each of the 3 time slices. So these are frequency distributions of the present-day poleward range limits in north or south latitude of lineages that we know from fossil evidence started in the tropics. Some stay in the tropics, the edge of which is marked in a general way by the vertical bar, but most get out. So we see the same out of the tropics pattern for lineages starting in the Pleistocene, as shown here, in the Pliocene, as shown here, and in the late Miocene, as shown here. And these results are really interesting for two reasons. First, the tropical origins are seen despite much poorer fossil collecting and preservation in the tropics. Dense vegetation and rapid weathering, and also just the greater intensity of collecting in temperate zones, at least to this point. So that means the 70-30 differential in origination must be an underestimate, probably an extreme underestimate. Second, the expansion out of the tropics has occurred in the face of the refrigeration of the poles, culminating the Pleistocene ice ages. So even as the high latitudes became increasingly harsher, this export process continued unabated, which is really kind of remarkable. So combining spatially explicit fossil data with the more detailed modern biogeography gave us a sharper picture of large-scale processes, although we still need to know, and this is really an open question, whether terrestrial clades follow the same dynamic. Okay, finally, as the last part of my talk, I want to talk about form. The partnership between paleontology and evolutionary developmental biology will be covered later today. So I want to take a different tack by asking, what are the dynamics of phenotypic and taxonomic or genealogical diversification? So here I'm just going to talk about the early history of a clade to the point where it reaches a maximum in one or both of those evolutionary currencies. What happens after that point is also very interesting, but that'll have to wait for another talk, unless Leo will give me another 45 minutes. It's often assumed that all major diversifications fit a single early burst model across scales and hierarchical levels. However, 3 end-member states might exist for this dynamic, as I'm going to show in the next few slides. In this figure borrowed from Gino Wesley Hunt's PhD work in Chicago's Committee on Evolutionary Biology, we have phylogenies above a tree, and notice they include both extinct and extant taxa, and below a graph of morphological variety or disparity and the number of taxa or diversity, both plotted against time. So type 1, shown here, is an early burst of morphological variety relative to taxonomic diversity. The Cambrian explosion of metazoans is the classic example. However you date the nodes of the metazoan tree, there's no denying there's a spectacular range of morphologies appearing over a 10 to 15 million year interval, a time span roughly equivalent to the history of cichlids in Lake Tanganyika, and only 2 or 3 times that of the anolis in the Greater Antilles. Type 2, the middle path, is the more general expectation, concordance between the two variables. Monty Slatkin, Michael Foote here at Chicago, and Bob Ricklefs each showed that simple diffusion in morphospace, a multivariate morphological space defining morphological variety, should be linear, and taxonomic diversification is exponential, and thus is linear on a log scale, and that's the key to the plot that I'll show in a moment for comparative purposes. And type 3, on the right, morphological diversity lags behind taxonomic diversification. Some type 3s have been called non-adaptive radiations, but extrinsic or extrinsic constraints on morphology could create this pattern, as could any diversification fueled by non-ecological speciation by Schluter's terminology. So using these 3 models, we can generate a diversity disparity plot, shown here. The horizontal axis is the log of diversity to create a linear expectation. The vertical axis is the overall morphological disparity of the clade, using, for example, Foote's variance metric, which is insensitive to sample size. Given data on a series of time slices through a diversification, and range standardizing them so that both axes range from 0 to 1, we can plot clade dynamics through time. If they fall in the upper left, they're a type 1 diversification exploding in morphospace at low taxonomic diversity. If they fall on the 45-degree line, the line of unity, morphological and taxonomic diversification are concordant, potentially prolific, but consistent with, for example, diffusion or Brownian motion. Or for that matter, concordance need not require time homogeneity, because if speciation pulses are accompanied by pulses in morphological diversification, the main point is concordance between the 2 metrics. And finally, clades in the lower right of the plot involve situations where taxonomic diversification, say numbers of species or genera as proxies, outstrips morphological diversity. Now, adequate data are still sparse for plotting more diversifications in this space, but some interesting patterns do emerge. Remember, each line is a time series starting at the origin and proceeding outward from there. I'll use a simple 2-point moving average to damp out some of the biggest sampling effects in the fossil examples, just for clarity, but the raw data, although noisier, show exactly the same pattern. So first we have a major segment of the early Paleozoic explosion, the blastozoans echinoderms studied by Michael Foote. And they do exactly what Michael said. They're well within the type 1 field, then calming down to converge on that line of concordance at the end of the time series. It's easy to imagine Jim Valentine's niche-filling models for the Cambrian explosion applying here. They're radiating out into a world lacking large metazoans, promoting prolific morphological diversification that eventually slows. In contrast, Kostov-Roy's analysis of aporeid snails, a major group in the Mesozoic, is a beautiful type 2 diversification, hugging that 45-degree line with amazing precision, something that neither he nor, I have to admit, I as his advisor, appreciated before recasting the data in this way. This also fits expectations from an ecological model. The clade is diversifying in the crowded world of the mid and late Mesozoic. It's making headway, but is a diffusive slog rather than an exuberant burst. Finally, there's the trilobites. These extinct arthropods also diversified the early Paleozoic, but as Foote recognized more than a decade ago, they're very different from the blastozoans. They go charging into type 3 space with numbers of species outstripping morphological diversity. Head up to and slightly overshoot the type 2 line, then parallel it. There are many possible explanations here, but it's hard not to notice that they're nested within the arthropods, which evidently do show a type 1 diversification. Cambrian arthropods and stem group relatives are so spectacularly divergent that they're actually almost impossible to plot on a single morphospace, which of course proves my point. Arthropods as a group explode morphologically, but the trilobite contingent within them do not. Where we do have both clades quantified at 2 levels, we can see that cross-level contrast. Here's the blastozoans again going up into the type 1 field, and one of their major constituents, the blastoidea within them, are more like the trilobites, keeping mostly to type 3 space with a quick late excursion to the type 1 relationships. Patterns differ across hierarchical levels. It's early days yet, but let's put these patterns in a larger comparative context. Again, there are few data available for quantitative terms to place them in a diversity disparity plot, but we can parse out a few qualitatively and start thinking about pattern and process. And here's a partial list of type 1 diversifications. Some of them are quite well supported by multivariate morphometrics, some maybe not so much. There's a cluster of early Paleozoic examples at the top, metazoans as a group, arthropods, echinoderms, that high-level extinct group, the blastozoa. There's vascular plants and tetrapods during the mid-Paleozoic, colonization of land. There's early Cenozoic teleosts right after the KT extinction, and a polyphyletic assemblage of carnivorous mammals, a guild, if you like, after the Cretaceous extinction too. But blastoidea within the blastozoans, arthropods within trilobites within arthropods, and carnivora within the carnivora morpha within the carnivorous mammals don't show type 1 diversifications. The dynamic isn't fractal. Subclades don't show type 1 diversifications, at least some of the time. And we do find type 2 diversifications after mass extinctions, like the cyclostome bryozoans after the huge end Permian event, and the carnivora after the Cretaceous. The big question, and we really don't really know this, is whether the insects, the angiosperms, the cichlids, the Drosophila, and others fall into this category. Oh, the textbook story is that they're all type 1s. The documentation isn't really there, and each of those radiations, though rapid and prolific, do show hints that disparity did not really necessarily outpace diversity. We just don't, but we really don't know that quantitatively yet. The early burst of species diversification inferred from molecular data for East African cichlids and Galapagos finches and Anolis lizards and Hawaiian honey creepers and so on is not readily equated with the discordance between taxonomic and morphological diversification seen in the classic fossil examples. They may even correspond to a different dynamic still. It's hard to say because we're missing all the extinct species in those diversifications. And by the way, I've got to say, given how much we know about Drosophila phylogeny and development, it's just nuts that a morphospace analysis hasn't been performed on the group and its subclades. So somebody get on that, please. Now, it's hard to compile a rigorously defined list of type 3 diversifications. of type 3 diversifications, where richness outstrips morphological variety. But who wants to measure 1 million similar taxa just to prove a point everyone can see with the naked eye? But there are the trilobites and a couple of their most diverse subclades, the huge snail order Neogastropoda, which was definitely radiating into a crowded world, the mammal clade Carnivoromorpha, the salamander genus Plethodon, disticid driving beetles with 174 genera, tenebrionid beetles, and so on. I'm sure everyone can think of an insanely species-rich clade that occupies an anomalously small range of morphospace. And given the pattern seen so far, we have to ask whether type 3 diversifications don't eventually transition to type 2s as species differentiate morphologically over time, a point in Price and Rodell's recent tree paper on 9 adaptive radiations. I can't resolve this issue today, but it raises a lot of interesting questions. I'll just say a few things. First, the presence of type 1 diversifications at times other than the late Paleozoic is another argument that even the most dramatic evolutionary bursts depend as much on ecological setting as on some aspect of developmental evolution. Obviously, these phenotypic changes involved evolution of gene regulatory networks, but these data do not compel an argument about the hardening of the genome after the Cambrian explosion, for example. Mind you, the Cambrian explosion was a unique evolutionary event, and there's much more to be learned about how developmental mechanisms themselves evolve over time. But I have to say, range standardizing morphospace probably damps out that aspect of macroevolution. And so other sorts of data should probably be used to address that particular question. Type 1 diversifications, the explosion of morphology relative to taxonomic richness, involves something special because morphology really does fan out at that higher rate, given a given taxonomic richness. But it's hard not to see ecological opportunity as the key factor. Remember the diversification in type 1 dynamics of the tetrapods and the vascular plants as they got onto land, for example, echoing, if you like, the Cambrian explosion in terrestrial terms. The question is ecological opportunity alone enough to generate the type 1s? The problem is that not every invasion into a new region kicks into a type 1 diversification. We still need a richer theory of evolutionary novelty to account for these differences among these dynamics. On the other hand, the perception that these are different types of diversifications isn't simply due to scale. For example, via the higher taxonomic ranks favored by paleontologists. We find different patterns among paleontological studies, sometimes within a single study at different hierarchical levels. And regarding time frames, remember the actual time scale of the Cambrian explosion, 10 to 15 million years, roughly the equivalent of Lake Tanganyika cichlid diversification. Even the Galapagos finches represent 2 or 3 million years of history at least. But the phenotypic action of the Cambrian explosion was only 5 times that. It took over 5 times that time interval. Give the finches 5 times more time in the Galapagos, and they'll still be finches. The great question, and this harks back to Doug Fatua's great talk yesterday, is what the finches could do with a genuinely empty world. It could be that we're seeing a difference between, or it could be that the difference between a type 1 and type 2 is much more sensitive to even a few representatives within these broader adaptive zones, so that a potential type 1 can transition to a type 2 with only a few taxa being present. And I'll show in a diagram what I mean in just a second, but the cross-level differences are really intriguing. I want to talk about that, remind you about that one more time. Arthropoda versus trilobites and so on. The blastozoans and the blastoidea, the mollusks versus the neogastropods and the Mesozoic carnivorous mammals versus Carnivoromorpha. It would be fascinating if this held up. And Wesley Hunt's data on mammals after the KT really may hold the key. The polyphyletic median gill appears to follow a type 1 diversification, while the clade Carnivoromorpha bounces between a type 2 and type 3 and never gets into type 1 space. Perhaps it really is all about open ecological space in some very broad sense. Meat eaters as a whole, one could argue, had a supercharged diversification fanning out through morphospace, but each clade had negative feedbacks, perhaps even at the outset, so the carnivora morpha show a more typical diffusive pattern. So the pattern of a type 1 diversification may actually look more like this, with a rapid spreading that establishes, I hate to use this term, adaptive subzones or individual clades, and then those individual clades get in each other's way even at relatively early stages in the diversification to produce a more diffusive or even a constrained diversification, even as the overall clade is moving quickly in morphospace relative to its total diversity. This is actually reminiscent of Harmon et al.'s 2003 paper suggesting that some diversifications first stake out adaptive themes and proceed from there, while others do not. And in a similar vein, both Valentine and Bombach, from different angles, have argued that the invertebrate phyla during the Great Paleozoic radiation largely corresponded to Simpsonian adaptive zones with relatively little functional overlap between them, with that overlap only increasing in a diffusive manner over geologic time much later. It'd be worth testing this idea more rigorously by adding a functional axis to the diversity plot, making it three-dimensional. But we haven't done that yet. So to conclude, for understanding large-scale evolutionary dynamics, including how short-term local processes scale up or fail to scale up, neither paleontology nor neontology has the whole picture. And the renewed partnership is breaking new ground. Neontology provides insights into genomes and developmental processes, ecology, critical aspects of phenotypes such as physiology and behavior, and there's a relatively complete sampling of species in a specific arbitrary time zone, the present day. The fossil record is imperfect, as Darwin was forced to claim, but perfection was never the expectation, just as we don't expect perfection in our experiments, our observations, and our inventories of living organisms today. The real issue is whether the record is good enough to tackle important questions rigorously and from new directions. And there the answer is surely yes. But the best work of all will involve the more complete integration of paleontology and neontology, with the aim of a richer understanding of evolutionary process and pattern. Thanks.